Hello friends, welcome to my channel Mediated Biochemistry classes. In this video, we will talk on the topic mechanism of splicing mediated by spliceosome. Okay, so how a spliceosome uh, affects splicing of spliceosomal intron, which is the largest class of intron in mRNA precursor of eukaryotics. So what you will find it that the mechanism of splicing mediated by spliceosome mechanism is similar to group 2 class intron self-splicing. So the mechanism of splicing mediated by spliceosome is similar to group 2 class intron self-splicing. I have already a video on it. Again, next thing is that the mechanism involved mechanism of what? Mechanism of splicing mediated by spliceosome of spliceosomal intron involve what? Well, it involves two things. First, it involves two trans-esterification reaction. Two trans-esterification reaction. Okay, and lariat formation. Lariat formation. Okay, so what is trans-esterification reaction? Trans-esterification reaction. What is trans-esterification reaction? Well, let us consider this reaction. That is, you have an ester, you know it. Okay, if you react with alcohol, so what is happening? You are getting this. So what is the specific reaction or a specific in the trans-esterification reaction? You can see that this organic part of ester has been exchanged by organic part of alcohol. Okay, so you call it trans-esterification reaction. So trans-esterification reaction involves exchange of organic part of ester with organic part of alcohol. Okay, so this is trans-esterification. And now lariat is, what is lariat? So lariat is, well, it is if you see in any structures, it will be like this. That is, you have a knot here, a knot here, and like this. So what is this? This is the lariat, and it is a rope used to catch. It is rather shape of rope used to catch animal or tiger. Okay, so these two things are part of mechanism of splicing mediated by spliceosome, that is two esterification reaction and lariat formation. as I told you about the lariated shape and what it means. So something like this is formed, rather lariat is formed, not something like a lariat type of structure is formed. Okay. Now, when we talk, we are talking mRNA precursor having spliceosomal intron removal. Okay, intron removal by spliceosome. And obviously, we are interested in mechanism because already we know it that this happens. Now, if we see mRNA precursor having a spliceosomal intron, what is the characteristic? Now, characteristic of what? Characteristic of this mRNA which undergoes splicing mediated by spliceosomes to remove a spliceosomal intron. It has been found that obviously, if you remember, you say this is axon, and then you say that it is junction, then you have intron, then you have again junction, you have this is exon. Okay, so this is your 3' you call it 3' splice site, and you call it 5' splice site. And what happened in a general splicing? The 2 exon get, if it is A and B, if you designate, so you have A and B exon joined, and removal of the intron removed. So this is your splicing. So what I want to tell you that in this is the general representation, you know it. But in case of this mRNA precursor undergoing a splicing mediated by spliceosome, it has been found that at 5' end, it has a specific dinucleotide that has been recognized as GU. Okay, obviously guanine and uracil, and at 3' you have adenine guanine. Okay, this dinucleotide is there, and you call it consensus sequence. Okay, you call it consensus sequence. S is missing. Consensus sequence. Okay. Now, apart from that, these dinucleotide is also their splice site. That is, GU is 5' splice site, and AG is 3' splice site. Okay, and this is part of intron. Okay, these are part of intron, and obviously these are exon. These are exon. Apart from this, you have from 10 to 40 nucleotide upstream. What you say? You say from 3' end, and from AG 10 to 40 nucleotide you have a branch site. Okay, and branch site contain adenine in it. Okay, you have branch site which contains a adenine in it. Now another interesting is this, apart from adenine, it has been found when you talk branch site, a typical branch site where in a spliceomal intron has been found to this sequence, that is YNYYRAY. You can remember it by simply yes, New York, Yak, Ray. So you will get it. Easily. The sequence at branch site has typical nucleotide sequence YNNYYRAY, and let me explain now. Y is what? Y is pyrimidine. Pyrimidine, you know, it can be cytosine or uracil. What is N? N is any nucleotide. N is any nucleotide. And what is R? R is purine. And you know, when you talk purine, you are talking adenine or guanine. Okay? And obviously you have this adenine. So adenine is must. It is loosely conserved. So you can see that you have a splice site. At a splice site you have dinucleotide GU, and at 3' you have AG, and you call it consensus sequence. So why consensus sequence? You also call it frequent sequence, because if it is not there or it is tampered or if it is missing, what will happen? Splicing will not take place. Splicing will take place, but it will causes inhibition. So this has to be there for splicing mediated by Spliceosome. Okay. Another thing, you can represent it like this also. That is branch site. I told you that it is Y. That is yes, New York, Yak, Ray. So you have, and this from 10 from 40 nucleotide. You have A, G, and then obviously your exon part starts 3' size, 3' end, and similarly you have up to here you have GU, and that is your these are all, you can say, branch site. If you want to remember, writing it again, no problem. And you have 5' end, and A, G, U, whatsoever you write, A, no problem. Better just okay. And these are the splice site. 5' splice site, and you have 3' splice site. Now you have three sequence: consensus GU, AG, which is frequent and changing results into inhibition, and you have this a bit loosely conserved but you have this branch site, which is a typical one. Now, when you talk about a spliceosome, you say that it is made up of snRP, which is made up of snRNA and protein. And snRNA, I told you, it will be U1, it will be U2, U4, U5, U6. So these three, how it will be recognized? This will be recognized by snRNA in snRNP, which is very specific for critical sequences. That these two sequences are also called critical sequence. Okay, because if you remove it or some disturbance, it causes inhibition. So now next is snRNA in snRNP. Okay, this gives a specificity to individual intron. What it gives? It gives a specificity to individual intron. Individual intron. Okay. So due to this specificity, what happens? This led to recognition of what? Recognition of critical splicing signal, where at 5 prime, 3 prime, and as I told you, and at branch site, or you also can say signal at 5 prime, 3 prime, and branch splice site of intron. So these sequences, consensus sequences, that is critical sequences, and branch site sequence in which adenine is there, has to be recognized by snRNA present in snRNP, and this is very specific, and it has very specificity as far as not for the bulk, but rather for individual intron. Okay.